Bismillahirrahmanirrahim. In this lesson, we are going to talk about the expression vectors and reporter genes. We had talked about cloning vectors. Expression vectors are also cloning vectors. However, they have the ability to express the cloned gene of interest. By expression, we mean they have the ability to make proteins out of those foreign DNA gene inserts. Now, these are very similar to vectors described before. However, in addition to other genes, they also have certain regularity elements in their sequence. For example, those regularity elements can be so many. The most important of those are promoter sequences on which RNA polymerase can bind and can start the transcription downstream. So if we have promoter region upstream of the insertion point of gene of interest in the vector, chances are that that inserted gene will be transcribed. So you can see here that it has different regulatory regions located before and after the foreign gene, which directs the host to translate the product of the of that protein, of that gene. Now, it is sometimes a difficult, expensive, or time-consuming procedure to analyze for product of the foreign gene. Why so? Because it is easy said than done. Expression vector is quite a tricky task. It takes a lot of time, lot of expertise, and lot of optimization. So how we can see the progress of our expression cloning? There are so many other ways and means. For example, you can see here there are certain marker genes which can be used to track down the progress of the molecular cloning experiment, especially when we want to do the expression of a certain gene. It is sometimes difficult, expensive, or time-consuming procedure to analyze product from the foreign gene, and so in addition to the selector genes described before, there are frequently reporter genes to indicate whether or not the signals are switched on, allowing the foreign DNA to be expressed. So basically, those are marker genes again, and this time they are being used to tell us whether our gene of interest has been started transcribing or not. Instead of waiting and looking at the end product of the transcription of that gene, we should have some signals before that just to see whether we are going on the right track or not. There can be so many different examples of expression vectors and so many different examples of the marker genes used in expression vectors. However, again, there are so many different circumstances that arise during the expression cloning experiments. Sometimes we can foresee those circumstances, sometimes they are just out of nowhere, and we have to optimize our experiments accordingly. There are many such genes in common use, and these are usually these reporter genes usually code for some enzyme of our antibiotic. We have talked about marker genes before as well. Reporter genes used in expression vectors are also such marker genes which can be used to track down the progress of our experiment. So again, instead of waiting for the completion of the experiment and waiting to look at the final product, we should have some indication before that, and that can be achieved by using these reporter genes. The reporter genes can be antibiotic resistant gene or any other gene of an enzyme, for example, even of beta-galactosidase enzyme, the example of which we discussed in our previous lessons. And if we use beta-galactosidase enzyme based vectors, the clones we get are either blue or white. White ones are the clones that we are looking for, and this type of screening, this type of indication, this type of tracking down of experiment is basically called blue-white screening of our clones. Now, there are other reporter genes as well, in addition to antibiotic resistant genes and enzyme genes, beta-galactosidase enzyme genes. For example, there are genes which can emit light. For example, luciferase enzyme from fireflies have been used in different DNA-based vectors. If we have such enzyme, we can check for the presence or absence of light from that enzyme. There is GUS gene as well, which is basically beta-glucuronidase enzyme, and it is also being used to identify the clones which are actually expressing our protein of interest and the clones which are not doing that activity. So you can see here there are so many different reporter genes which are used in expression cloning experiments. Now, a thing to remember is that these reporter genes, just like any other marker genes, they are just an indication that what is going on. They just tell us whether the transcription is happening or not, whether the translation is happening or not. They do not tell us how good our protein is being made, how much our protein is being made. As with selected genes, the reported genes serve no useful purpose once the cloning procedure has been successfully accomplished to produce the finished product. So they are just an indication for us to assess that whether our experiment is being performed correctly or not. So once we have completed the experiment, only then we can look at the final product, which is our protein of interest. This was about the expression vectors and the reported genes.